Thank you so much for the invitation and for this very elaborate webinar and recording today. Anyway, thank you. So as you probably have realized from the introduction, my research program is pathogen agnostic. I'm really more interested in investigating infectious disease pathogenesis and how therapeutics can help to protect pregnancies. And we're getting closer to figuring out a few things about influenza virus disease pathogenesis and its impact on pregnancy and many other things along the way. And so I thought that this would be a good story to tell this department. Within the past 2 years, I've had a financial relationship with GlaxoSmithKline as a consultant related to an RSV vaccine trial. This is the team that did all of the experiments, did all the work that I'm going to show today. I also wanted to highlight Orlando Cervantes, who is a pathobiology PhD student, and nearly all the work that we're going to show today is part of his thesis, and Jeff Munson, who also assisted with the biostatistical modeling that you'll see today. But everyone on that slide helped with these experiments. So influenza A viruses circulate globally. They're always a pandemic level threat. There are 4 types of influenza A viruses, and these are the ones that cause pandemics. Subtypes are classified based on the combination of hemagglutinin and neuraminidase, which are the blue and red little molecules on the surface. There are currently 2 subtypes of dominant seasonal viruses, H1N1 and H3N2, and I'll talk a little bit more about those. There's also a highly pathogenic avian influenza subtype, H5N1, that you probably have seen in the news recently, and it's an emerging zoonotic disease threat. We'll talk more about that. And here you can see the hemagglutinin protein that the virus uses to attach to the cells, decorating its surface in blue, and the neuraminidase, the N, which enables the virus to be released from the host cell. And that's why it's called H and N. It's just different types of these proteins. So influenza A virus pandemics by subtype, there have been many over the last century or so, and they keep evolving and keep changing. And there are new avian influenza viruses that threaten to combine with these old ones to make new ones. And so I kind of like this graphic very much. It shows how we start with H1N1 influenza virus, and then what happened in 1957 is that we took H2N2 from an avian virus, recombined with an H1N1 human virus, giving you an H2N2 that then could be transmissible in humans. In 1968, we had the Hong Kong influenza, when we used to name everything by the place where it came from. And we had an H3 avian virus that recombined with an H2N2 human virus, making it transmissible among humans to make an H3N2. And the seasonal viruses that we have are related to these, the H3N2 and the H1N1. But now we're a little bit more worried about H5 bird flu. And so for the next pandemic influenza, either it's going to be an avian virus that simply mutates and is ready to go and then becomes transmissible between humans, or the avian virus will combine with an H3N2 or an H1N1 and create a brand new virus that is transmissible to humans. H5 bird flu is really widespread in wild birds in the US and globally. It's caused outbreaks in poultry, sporadic infections in mammals, and right now there's a big outbreak of H5N1 in dairy cattle in the US. And recently 2 reports of cases in humans. However, there was an H5N1 virus infection in farmed minks in Spain in October of 2022 that was really very concerning. And that's because, well, first they found minks that were really ill. About half were dead in the pens. They figured out that it was H5N1. The problem was that it had acquired an uncommon mutation in one of its proteins, and that same mutation was present in the same gene in H1N1, and that mutation could then lead easily to another mutation that makes it transmissible, to make it confers human receptor recognition. So it was kind of one mutation away from being able to have human receptor recognition. So a very close call, and people on these mink farms, they weren't wearing masks, they weren't kind of doing anything to mitigate this problem. So bird flu is definitely edging closer to human spillover. So the blue states here are where H5N1 has very recently been detected, either in dairy cattle, in goats, in cats, so a number of different places. So CDC now has a current situation summary that you can access every week to get a better sense of what's going on with H5N1 bird flu. So you can see that it's widespread in birds, flocks, mammals. 2 cases in the US, and the current public health risk is low. But I don't think they would have a place on their website if they weren't concerned. So antivirals may be effective. They say candidate vaccines do exist, but time is always needed to produce more vaccines for the entire population. And seasonal influenza vaccination is not effective against H5N1, and it's highly pathogenic. So what's a better plan? Well, a universal influenza vaccine would be a much better plan. And the NIAID came out with a funding announcement in 2019, which was just before the COVID-19 outbreak, which kind of turned everything kind of on its end. And it was about this time that I became very interested in influenza, because NIAID wanted to understand its transmission, the natural history and the pathogenesis of influenza infection, characterize how protective immunity works, and how to tailor vaccination responses to support the design of universal influenza vaccines. And you ask, why is an obstetrician interested in this? And that's because for a universal influenza vaccine group, it needs to be protective for all age groups and vulnerable groups, and pregnant women are one of those groups. So a universal flu vaccine needs to be at least 75% effective. It's got to protect against both group 1 and group 2 influenza A viruses, should last more than a year, and really cover a wide portion of the population. And so to do that, you need to target your vaccine to epitopes in the stalk of the virus. And that's that kind of bottom part. If you think of it like a mushroom, it's the stalk of the mushroom. That's where you've got those conserved epitopes that could lead to generation of a universal influenza vaccine. And it seems like things are getting closer, although COVID-19, I think, did throw a wrench in the whole research pipeline. But here are the vulnerable groups that need to be protected by an influenza universal vaccine. And pregnant women are one of those groups, and they're always harder to study. They're harder to convince that there is safety in vaccination. And pregnant women from influenza have higher rates of preterm birth, stillbirth, and maternal death. And they had very high rates of maternal mortality in the 1918 H1N1 pandemic. In 1957, if you just looked at all the deaths in reproductive age women, about half of them were pregnant. And in 2009, there were a lot of hospitalized women, intensive care, 8% died. I was pregnant in 2009 myself, and people kept calling me, telling me about pregnant women with influenza dying in different ICUs across the state and in neighboring states. Very concerning. So there is a disproportionate impact of influenza on pregnancy, as there are for many, many other infectious diseases. And there's also some concern that it could infect the placenta. And the reason for this is that human influenza A viruses use an alpha-2,6 conjugated sialic acid as an attachment receptor, which is really abundant on the upper airway epithelial cells. But that's not the only place that has these alpha-2,6 sialic acids. They're in other organs: brain, liver, kidney, heart, intestine, and if you can believe it, in the placenta. And so there is something called TORCH, which is something that most medical students learn along the way. It's an acronym really for all the different pathogens that can infect pregnancy: Toxoplasma gondii, syphilis, varicella, rubella, cytomegalovirus, herpes simplex virus. And as time has gone along, we found more and more pathogens that can be added into this group. And so the question is, could influenza also be a torch pathogen if it could possibly infect the placenta? There is a little bit of evidence that it can infect the placenta. It can infect placental explants, and it has been shown to do so just with immunohistochemistry. So kind of at a very shallow level here. There's also concern about influenza and what it does to the developing fetal brain. And this started when there was a big population study in the Scandinavian countries, starting in Finland, where they had a very large finished birth cohort. And they looked to see during the H2N2 pandemic, they looked at all of the psychiatric hospital diagnoses for all individuals in Greater Helsinki, and then figured out how many of them would have been fetuses in the second and third trimester during that H2N2 pandemic. And they calculated there were higher rates of schizophrenia in those adults who were unlucky enough to be fetuses during that time. The same type of study was also done in Sweden. We actually were part of that study. We looked at about 2 million people that were observed from birth up to age 41 years. It was more than 32 million person years. We linked ICD-9 codes for the mother and the child. And during part of it, my student, epidemiology PhD, he threw away half the data as he was modeling and testing things, so we would do it. Had he not done that, schizophrenia would have been significant. But even so, we had high hazard ratios for the inpatient diagnosis in a child of autism, as well as depression, as well as suicide, for fetal exposure to any maternal infection. So it's not just influenza, it's many other infections as well that we think can probably alter fetal neurodevelopment that can then manifest decades later in a higher risk of neuropsychiatric disease. So this is my background, this is where I come from. There's just something so great about delivering a baby. So the questions that I have when I take that lens and apply it to this literature is, why on earth would a young and usually healthy pregnant woman be predisposed to severe disease? There has to be a reason for that. And there has to be some sort of host response or immunologic defect or something like that. And the prevailing rumor and theories, which are based upon nothing, is that it's either a cytokine storm or it's some sort of immunosuppressive reason that we haven't figured out yet. It's just pregnancy immunosuppression. The other question I have is, how does flu impact the placenta and fetal development? We should know this much better than we do. If it does have if it is associated with a high rate of schizophrenia, we should know how that works and whether we can impact that and prevent those problems. So I thought it'd be a good idea to create a new pregnancy model of influenza A virus, and I had a lot of questions when I was coming up with that. First, related to lung disease, what's the best translational model of respiratory viral infections? How do you best model pregnancy physiology in particular? And are we measuring the right disease correlates to enable uncovering of differences in pathogenesis between pregnant and non-pregnant individuals, and maybe when those differences are kind of subtle? I also had questions about the placenta. Will the animal model enable us to determine the impact of flu on the placenta and fetal injury? And what's the best animal model of human placentation? And so comparing across different animal models that have been used for influenza A virus disease, ferrets, mice, guinea pigs, swine, non-human primates, and zebrafish, there are a lot of issues with different animal models that make them less translatable to human pregnancy. And so the non-human primate is the closest animal model to human pregnancy, and one that I've used for many years. But there have actually been limited efforts to optimize influenza non-human primate models, if you can imagine. I don't know why it hasn't been as well developed as it should be, but there had been relatively few models in the past, and then when COVID-19 came, everything turned to COVID-19. But even so, the COVID-19 disease models in non-human primates, in my opinion, were not as well developed and modeled, they were done quickly to try to test vaccines, but in a less careful way. Mouse models are not ideal to study pregnancy for lots of reasons that I'm not going into, but there are many, many differences. And non-human primates have a long singleton gestation. Their reproductive biology is very similar to human pregnancy. And you can determine maternal fetal injury, vertical transmission, and whether new therapeutics can protect the fetus. This is my lab. These are kind of some of the things that we do. And as I talked about, I'm kind of pathogen agnostic. Whatever the new pathogen is, I want to use it and figure out how it works in pregnancy and therapeutics. And there's some very basic program objectives that we have that we try to translate to each of these new pathogens. First, how do they suppress or overwhelm host immune responses in the mother, the placenta, and the fetus? So it's like triple the work. You can't just figure it out in one, you have to figure it out in all three. What are the immunologic correlates of disease? What are the mechanisms of injury? And then can we test new therapeutics to prevent maternal disease, preterm birth, fetal injury, and so forth? So with influenza disease, the first question was, what are the correlates that might highlight subtle differences between pregnant and non-pregnant state? And there's got to be something we can do to measure pulmonary physiologic measures and immunologic correlates. And we really have to do both to kind of get a handle on what's going on. So this had been kind of the gold standard of measuring pulmonary physiology. It's a plethysmography chamber. And in it, you can put an animal, and as they breathe in and out, the variation in box air pressure and volume is measured. The volume of air shifts around, and they can calculate tidal volume and functional residual capacity. Well, that's great, but you can't assess gas exchange and pulmonary mechanics very well. And if the animal is ill, macaques need to be anesthetized to go into a chamber like that. And it's not really good if they are, if their respiratory status is compromised. And in pregnancy, lots of things change. So we need to find some way to measure this in a much better way, especially oxygenation. The other thing that flu does is it activates and recruits many innate and adaptive immune cells. So we need to look at this with really broad strokes. And these are just some of the potential immune cells that can be changed and might be responsible for differences in pregnancy disease pathogenesis. So one of the first cells that we got interested in were cell types that express IL-17. And several publications had connected IL-17 in either resolution or enhancement of influenza infections. So if you have too much or too little, that can be a problem. So early production of IL-17 by gamma delta T cells promotes viral clearance, really important. If you don't have that, you can get a prolonged disease course. Also critical for the B cell response. However, if you have too much production of IL-17, it can induce immunopathology. So it's kind of like the Goldilocks hypothesis. You have to have just enough, not too little, and kind of not too much. Well, in pregnancy, kind of interestingly, both estrogen and progesterone, which are higher, drive down ROR gamma T, which is a critical transcription factor for Th17 cells, which are T helper cells that express IL-17 and IL-22. And there are lots of epithelial cells that have IL-17 receptors at mucosal surfaces, including the lungs. And so this is really important in producing chemokines and neutrophils and antimicrobial peptides. But in addition to Th17, we've also got cytotoxic TC17s, and we've got gamma delta T cells that express IL-17. So none of this can be easy. It all has to be very complicated, and there's not just one cell type that we're interested in, there's three. But actually, there's more than three. There's probably 40. But we're gonna focus on just the one here. So our hypothesis was that there was an aberrant balance of IL-17 expressing cells, either Th17, TC17, or gamma delta 17 cells, and maybe even T regulatory cells early during an influenza A virus infection, and that this predicted pathological correlates of enhanced influenza disease in pregnancy. And we thought that a pulmonary physiologic measure would be best. We also hypothesized that flu infection could lead to placental infection, maybe even vertical transmission to the fetus, and a pro-inflammatory antiviral response that predisposed to fetal injury and stillbirth. This was the study design that we came up with. Day 3 is a very important time point for Th17 cells, so that was absolutely critical that we measured that. We had a 5-day time course, which we wanted to look at really early correlates of disease, and at 5 days we did a cesarean section, a Diamond fetal necropsy, or if the animal was non-pregnant, which was the other half of the group, we just did a necropsy at 5 days. And we thought we would find peak pathology there. And certainly some animals were extremely ill and couldn't have gone on another day. We did pulmonary function testing, both at baseline and at day 5, and we did bronchoalveolar lavage, fluid sampling at baseline, again at day 3 and day 5, because day 3 was such an important time point. The first thing we had to do was to figure out how to test pulmonary physiology in a way that would yield a lot of quantitative metrics that we could then use to model all of our data. And I did the same thing that I always do as a physician. When I don't know what I'm doing, I start calling up people and calling in consults. So I called Bill Altemeyer from Pulmonary and Critical Care Medicine and said, I don't know what I'm doing in this area. What would you recommend? And he said, well, in humans, this is what we would do. We would anesthetize them, we would ventilate them, we would administer a paralytic agent, we would wait until they were not spontaneously, they had no spontaneous respiration, we'd collect a blood gas, and then we would perform an inspiratory pause. That would allow us to measure the pressure plateau, the pressure peak. From there, we could take all those metrics and give 2 outputs of oxygenation, which is the partial pressure of oxygen in the arterial blood, divided by the fraction of inspired oxygen, and the alveolar arterial oxygen gradient, and as well as lung compliance. And this seemed to me to be a great advance to what had been done in the rest of the field and would give us a real chance to try to figure out pathogenesis of disease. It took quite a few people. And these were the metrics on the left that we were able to obtain. And on the right, you can see peak airway pressure, plateau pressure, this inspiratory pause that was really key to enabling us to calculate lung compliance. And so here is some of our data. I just want to check, yeah. And the dashed line, beneath the dashed line is the threshold where acute respiratory distress syndrome is diagnosed in humans, partially diagnosed in humans, with a less than 200 value reflecting severe hypoxemia. So we've got a baseline measure and a day 5 measure, and you can see that they go down in essentially all cases. The slope of the line is similar for many of them, but in some of the cases, it's a very, very steep decline. The blue lines are non-pregnant, and the red lines are pregnant. So you can see some of the steepest declines happened in our non-pregnant animals. We've actually got a mix of both non-pregnant and pregnant animals below that dashed line. So we really had a mix of severely ill animals, both pregnant and non-pregnant. It wasn't so binary that only pregnant animals became ill. And this was a highly significant change from baseline to post-infection, as you can imagine. But there was no overall association between pregnancy status and disease severity, just by looking at this first graph. Next, we checked whether we had viral RNA in organs, both by quantitative PCR and by plaque assay, and we did, as we expected. We found it in the maternal heart, maternal lung, lymph node, and we found replicating virus. So certainly the non-human primate was permissible to the H1N1 strain that we inoculated. We found extrapulmonary disease. So all the animals had evidence of a pneumonia, but we also found extrapulmonary disease. So here's a pregnant animal that had myocarditis, brain and spinal cord injury, and this is staining for CD3 with T-cell infiltration. We asked our pathologist to use a lung score that was developed here in the lab medicine department, and they were blinded to the groups, And what we found is that we had higher lung pathology scores in the influenza group versus the controls, so pathology was definitely achieved. But there was no significant differences between our pregnant or our non-pregnant group, although there might appear to be a trend to having slightly higher scores in the pregnant group, but that was not significant with our numbers. Next, we looked at our pro-inflammatory cytokines in the pregnant lung compared to the non-pregnant lung. And here we had no significant differences, but it looked like there might be a trend to having higher cytokines, some higher pro-inflammatory cytokines in the pregnant versus non-pregnant lung. But clearly there's a lot of overlap there between both groups. So next we asked Dr. Deborah Fuller and the talented scientists at her lab to help us with a flow cytometry and intracellular cytokine staining panel so that we could really look very broadly at different immune cell types, and specifically at cells that expressed IL-17, either Th17, Tc17, or gamma delta 17 T cells, as well as Tregs. And the first thing we were really interested in was looking at the change in Th17 in peripheral blood mononuclear cells over time. Again, blue is non-pregnant and red is pregnant. And the peaks you see here at day 3 is what we kind of had expected for Th17. Those 2 peaks in the 2 non-pregnant animals were associated with the most significant disease in our study in the non-pregnant group. Interestingly, in the pregnant animals, only one of them had that peak, and they seemed to kind of be lower, both mainly at day 3, but then a little bit higher at day 5. Neither of those changes were significant. We looked in gamma delta 17 cells in the pregnant lungs in day 5, and we had no difference. This was in the airways from bronchoalveolar lavage as well as in the lung. And on day 3, we had no differences, but on day 5, we had very, very low gamma delta GD17 cells in the bronchoalveolar lavage and in the lung compared to the non-pregnant animals, which is what we would have expected from our hypothesis and from the high estrogen and progesterone state. We also looked very, very broadly at other cell types and modeled them using our oxygenation metric, the PF ratio, on day 5. And I was really very interested at what is going on day 3 that predicts what happens on day 5. And we found that in both the pregnant and non-pregnant groups, differences in some of the frequencies of these cells were significantly correlated with the oxygenation status at day 5 for classical monocytes at day 3 and plasmacytoid dendritic cells, which are big interferon producers at day 5. So next we wanted to kind of model this and get a sense of altogether what are we seeing. We're seeing some significant findings here and there, but trying to put it all together and see what is really driving this disease. So we just did a multiple regression model because fortunately we had kind of quantitative variables for things. And in the first model on the left side of the screen, we're really just looking at the effect of pregnancy status on the oxygenation PF ratio at day 5. And pregnancy status had a p-value of 0.07. It explained about 17% of the variation in the model, which was the R-squared. If we added age of the animal and weight of the animal, we know that obese individuals have more severe disease. Suddenly that increased, that explained more of the proportion of variance in the model to about 54%. And in this model then, pregnancy was significant and age was significant. And both age and weight are correlated with each other, unfortunately. The effect of classical monocytes was something that we looked at as well for day 3 in the bronchoalveolar lavage, just kind of as an example to see what would happen if we added in one immune cell subtype. So here we have a very simple model on the left with pregnancy status, and pregnancy status is not significant in this smaller model. But then when we add in the classical monocyte frequency, we get, and pregnancy status, it's a 0.09 p-value. So it helps to improve the proportion of variance just with a single immune cell subtype. But now let's take a look at it with age and weight. And so on the left side, we've included pregnancy status and age and weight. When we include all of those together, we can explain 91% of the variation in the oxygenation status at day 5. When we now include classical monocytes into that model, it is no longer significant because pregnancy and age alone explains so much of the variation. It's very hard for any immune cell subtype of any kind to explain more variation. I find this hard to believe as someone who studies immune pathogenesis that we can't find something that will help to explain and improve this model. So we've thought about different modeling techniques and possibly lumping in pregnancy, age, and weight and making it a single variable, and then on top of that layering different immune cell subtypes. But it has not been an easy answer trying to understand this immune pathogenesis. But certainly we're in a very good position to test therapeutics in a universal influenza vaccine. The next part of our study was to look at the impact of influenza A viral infection on the placenta. And to our surprise, we were able to detect influenza A viral RNA in placental tissue using quantitative PCR. And we've also done TCID50 assays, and in one case, that's also been positive, showing replicating virus in placental tissue. Next, we looked at just a 14-plex cytokine panel, and we found pro-inflammatory immune responses in the placenta from the infected animals, significantly higher IL-1 beta, IL-18, GM-CSF, IL-6 as well, and TNF-alpha. But you can see that those have one big large outlier, and so if we remove that outlier, even so, we still have a significant difference between the pregnant and the uninfected groups. When we did a total RNA-seq experiment to look at placentas from cases of maternal H1N1 influenza infection versus uninfected controls, we see an incredibly strong type 1 interferon response in the placenta. All of the genes that you can see on the right-hand side both have a significant p-value and a log 2-fold change that are above 2-fold. And you can see the interferon-stimulated genes. Almost every gene on that right side is either an interferon-stimulated gene, an ISG, or a viral sensor that's being upregulated. This is the strongest type 1 interferon response that I've seen of any tissue that I've looked at over many years. So is this good? Is this bad? Is the placenta helping in the infection? Is it, what's going on? Maybe it is helping. Maybe it's helping to protect the fetus from an influenza infection. However, if you get a lot of type 1 interferon signaling, you can get stillbirth in a mouse model of Zika virus infection. So it inhibits development of the placenta. So type 1 interferon signaling is not ideal in the placenta at all, and that's what it wants to do after influenza, which probably is the link between influenza infection and stillbirth. So I think influenza A virus should be added to our idea of TORCH. There's just so many infectious diseases that can transmit to the fetus and cause fetal injury, and there are just too many to be captured by a single acronym. And so understanding how this works has been a very, very interesting career. So just a few conclusions just from the lung side of this talk. Pulmonary physiology testing is really ideal to understand the heterogeneity of the lung disease, measure it as well as we can, and be able to correlate with a number of different other quantitative variables. Influenza A virus viral load qPCR provides evidence of systemic cardiopulmonary infection in non-human primates as well as potentially placental infection. High Th17 frequencies in PBMCs that absolutely correlate with severe disease in a few non-pregnant animals. But this is not the case in pregnancy. This is not how that works. They're very low and suppressed. We've got lower CD4 positive gamma delta 17 cells in pregnant versus non-pregnant bronchoalveolar lavage and lung at day 5, which may impair flu clearance in later stages of the disease. So the question is, if we took the model out another 3, 4, 5 days, would we see a greater difference in some of these immune cell populations? We've got Th1 and pro-inflammatory cytokine concentrations that are very high in the placentas of the flu-infected non-human primates and a very strong type 1 interferon response signature in those placentas. And I think influenza A virus qualifies as a torch pathogen in that it transmits to the fetus, and we have evidence of that, and has been associated with this higher risk of late onset neuropsychiatric disease. So thank you very much. And this is how you get credit for attending today's lecture. So thank you. Steve? Go ahead.
For the Zoom audience, do I need a mic or anything, or no?
I don't think so. I'll repeat
Okay.
the question.
Okay. So first of all, it makes me rethink the meaning of a torch song. And Zika needs to be on the list too, right?
Yeah, for sure.
Yeah.
Yeah, Zika's on the list.
So what's the current thoughts about, like, I don't know anything about influenza and pregnancy, about how influenza, a respiratory pathogen, gets to the fetus? Because like we've seen with SARS-CoV-2, it can get into the GI system, you cough it up a bit and maybe swallow it and it gets in there. But it can go, SARS-CoV-2, COVID is in your lungs, but it can get into other tissues. It can get into the liver. But I just wonder what your thoughts are about, or is there a dogma for how, or a mechanism for how it gets to the fetus? Does it go systemic?
COVID-19?
No, sorry, flu.
Flu.
Yeah.
Yeah, flu is hard to detect in plasma, but it certainly must get around because we detect it in many, many other organs. We can get replicating virus. We can get it out of several different organs. And the placenta is full of receptors, and the placenta becomes infected. It's just hard for us in our models to really get it, find it, get it from the blood and from the plasma. But COVID-19 is a super interesting disease in its own right. For both flu and COVID-19, the impact that it has on the placenta is probably the greatest threat to pregnancy of all, because once it starts to impair placental development and placental function, then the fetus is at risk for stillbirth or impaired neurodevelopment. It doesn't even need to go to the fetus for that to happen. So a lot of myths around, well, if it doesn't go to the fetus, then it shouldn't be so bad. And I don't think so.
Is it known for COVID how the virus is getting to the fetus?
There's a lot of debate in the beginning because the transcriptomics data didn't show a lot of co-expression of the receptor and its co-receptor in the placenta. They looked at many different stages. But yet, with more and more evidence, it found that it could infect the placenta, just not at very high rates. And there were a lot of case reports trying to prove that the fetus had become infected in utero and not postnatally. And it can happen. I think that it's rare, but most importantly, it's the impact on the placenta and how that can indirectly impact fetal development. That's the bigger threat.
There is a question here that I can read to you from Chihiro Morishima. Are the immune cell markers you listed in a single flow panel, have you considered looking at myeloid-derived suppressor cell levels in your model? Most of the MDSC markers are in your panels.
We have done a number of different combinations. I cannot say that we have exhausted all of our analyses. Please send me an email. I'm easy to find in the system. We're still looking at everything, and it's a huge amount of data. We've looked at our kind of first hypothesis. It's not so easy. It's not so easy, and especially with our model and so much of it being explained by pregnancy status. My question is, should we even have pregnancy status in the model? Is that kind of pushing us too far to explaining all of the variation? So we're rethinking our model. We're adding in every population that we have, and we have a very, very big panel from all of these things to kind of look at it. But it has not been so easy to solve.
Well, and Chihiro maybe can get in touch with you.
Yeah, absolutely.
Chat about that. Yeah. Are there other questions? Because I have a question.
Yeah, go ahead.
So, most pregnant women who would get infected with flu would have been vaccinated or had a prior infection at some point. So they would have some pre-existing immunity. So of course, my question's going to be about B cells, but have you ever looked at the repertoire of B cells against a pathogen, or just the repertoire in general during pregnancy, like a baseline, and then see what happens when, during pregnancy to know whether that could be immunosuppressive or,
Yeah, that is a really, really great question. Some of that has been done in Harvard for, with the COVID-19 pandemic. They had kind of big cohorts that they looked at vaccination. We have not done that because we're just kind of figuring out how this works. We've done a little bit with CyTOF too, to look at just a very, very early kind of markers of interferon response and NK cell activation. And there seem to be some differences there with vaccination in pregnant versus non-pregnant individuals. But I think that there's still a lot to know, a lot to learn.
For sure. Yeah, I would be happy to talk to you.
Yeah, it sounds good.
Other questions, or I'm not seeing another question come up in the Q&A. Does anybody in the audience have questions? No?
All right, thank you so much.
Thank you so much.